Hi, ladies and gentlemen. Welcome back to video 2. Apologize, the lighting is so terrible, but you don't actually need to see me because we're going to look at a PowerPoint together. So let's take a look at transcription. So as we mentioned earlier, we are talking about transcription today. Transcription, and before I even get started, sorry, you're going to need your notebook, pencil, or a pen or a pencil. So make sure you get that. Pause me if you need to. And so again, we're thinking back to yesterday, we talked about, well, why do we even need this RNA as the intermediate step, or that step in between turning DNA into the protein, which is expressing a trait? And so let's jump straight into this. This should look familiar. It's exactly the same picture as yesterday from the central dogma theory. The idea that DNA is turning into RNA, which is turning into proteins. And this DNA and RNA, we call this our genotype. Basically, it's the instructions in our DNA or our genes. It tells my hair to be brown or my eyes to be brown, in this case, my example. My phenotype is the physical expression, so it's what we actually see. In mine, you see brown eyes. In my mom, you see blue eyes. In my brother, you see green eyes. So what we see is our phenotype. Our phenotype is dictated by what's in our genes. And so today, we're actually going to focus on this arrow right here. So how do we go from DNA to RNA? And that step is called transcription. So transcription is step 1, which we're going to learn about today. Our next step will be tomorrow, which will be step 2. But today we're focusing all on this star. So if you want to go back to your central dogma drawing from yesterday, where you wrote this out, and label this one transcription. So step 1, transcription. And so transcription, as we can see in this picture, takes place exclusively in the nucleus of our cell. So we need our DNA. The DNA is only found in the nucleus, so will be found during transcription in this piece. Translation is step 2, and that is tomorrow, and we'll talk about that, how it happens outside. So just a couple things. Most of you already figured this out based on that first part of class where you did, you looked at the model, but the location is inside the nucleus. So the entire step of process of transcription takes place in the nucleus. The enzyme involved is RNA polymerase, and as always, it ends in ACE, so you should know it's the enzyme. The monomer, so what are the building blocks? The building blocks of transcription are mRNA nucleotides. The A's, the U's, not the T's, the C's, and the G's. And the polymer that's created is our mRNA. And I keep saying mRNA, and what I mean by M is messenger. It's the messenger RNA. And it's kind of nice to think about it as mRNA or messenger RNA because it's taking a message from the DNA, it's copying it like a photocopy, and it's carrying it somewhere else. So it's carrying a physical message of what the protein should be. Before we get into steps, I'm going to try this. I'm going to share a new screen with you. Okay. So I'm going to draw this out because I know some people like this visually. If you are not a visual person, I will go through it step by step after, but we'll continue this drawing tomorrow. So if you want to have it take up an entire page like I am on your notebook, that's great, and we can add on it. So we'll start with our nucleus. And so our nucleus, I'm going to label it nucleus. Our nucleus is where our DNA is. And so I'm going to draw a partial double helix, one that's starting to unwind because we are going to use it. So this is my DNA. And so the DNA has one strand, and so I'm just going to say it's this strand, and this is our template strand. It's what we are going to copy. And so our RNA is going to come in, and it's going to make a very long copy of a specific gene. And that enzyme, RNA polymerase, is going to add those bases. So this is my mRNA strand. And these are all, I'm not actually going to write letters because it'll take a long time, but that's our mRNA strand. And so the RNA polymerase adds these RNA nucleotides. It makes this long strand, and eventually it will free itself. It will kind of be floating through mRNA until eventually it will leave the cell. But for this part, step 1, transcription, we are going to be exclusively inside the nucleus. And so that's what you need to have down for transcription, step 1. We know what happens inside the nucleus. We know that we're using the DNA and we're turning it into RNA. And we know that this is our end goal, is to make that mRNA. So if you need to pause me, pause me. And now, for those of you who are not my visual learners, which is very much okay, I want to show it to you in a different form. So this is the step-by-step instructions in words. So the DNA unwinds and the bases break apart. All of this is in the nucleus. The template strand of the DNA matches up with the complementary free-floating mRNA nucleotides. The mRNA detaches from the DNA, and the mRNA leaves the nucleus to go to the cytoplasm. So that's the same thing we just drew, but just in words. So if you want to have it both ways, both in words and drawings, that's great. If you're not a drawer, you don't have to draw. I would suggest it though, just to get like a general idea of where the different things are happening. And so that's transcription in a nutshell. The goal to make that mRNA, so then we can make that protein. We're going to try something. I have my .cam set up, so I'm going to show you how to do part 2, the first part of it. You can pause me, go back and forth between the two, have them side-by-side screens, whichever one works best for you. But we're going to jump through apply it part 2, which is in the worksheet. All right, so if you were to look at your worksheet on Google Docs, it will look like this. Part 2, apply your understanding. And the first one is this table. It says, DNA replication is the process by which a cell copies its DNA. DNA replication, during replication, both strands of the double helix are used as templates to make complementary or matching strands of DNA. DNA transcription is the process by which a single strand of DNA is used as a template to generate a strand of mRNA. The template DNA provides the instructions for the mRNA, and that's really important. There are 2 strands in DNA, but we are only focused on the template strand. Fill in the missing information below. So I completed this one for you, so I'm going to walk you through this one and then 2 others, and then you'll go through and practice on your own. So our template strand of DNA, this is our strand we're using for our mRNA. So we're using this template strand to match with our mRNA. The complementary strand of DNA is just strand 2. So we'll call this one strand 1, and this one strand 2. If you'll notice, this just follows our normal Chargaff's rule. T matches with A, T matches with A matches with T, C matches with G, and G matches with C. When we get over to mRNA, we're noticing that, okay, so again, T matches with A, same thing, T matches with A. But now we have A, instead of matching with T that does here, it's going to have a U. So that U is new. And then C matches with G, and G matches with C. So if another way to double check is that complementary and messenger are essentially the same. If you look here, the only difference is anything that's a T here is going to be a U here. So this is still AA, AA, U instead of T, and then GC, GC. So it's an easy way to check. I'm going to walk you through another one. Let's look at the second one. We have our complementary strand of DNA. So we're going to first find our template. So we have GG. So what matches with GG? Well, CC. Then we have CGG. Well, C matches with G, and then GG would be CC. That's our normal Chargaff's rule. Now we need to find our messenger RNA or our mRNA. To do that, we first figure out that, all right, messenger RNA, we need to look at our template strand. What matches with CC? Well, G. All right, now what matches with GCC? Well, again, G matches with C, and then G. You'll notice that these two are identical. GGC, GG, GGC, GG. Because there's no T's, it's going to be exactly the same. So that's your template strand. Sorry, that matches with your template strand, but it's also the same as your complementary strand. This isn't always the case because, remember, when we have a T, it's going to turn into a U. Let's jump down to this one right here, AGACTC. Template strand to complementary, normal Chargaff's rule. A matches with T, G matches with C, A matches with T, C matches with G, T matches with A, and C matches with G. So that should look pretty straightforward. Our messenger RNA is going to look a little different this time because we have a couple T's in here. A matches with T, but because we're an RNA, we know it's a U. G going to match with C, A, U, C, G, T, A. That's a biggie. A lot of people think that this one's going to be a U too, but because it's an A, A's are also an RNA. And then finally, C is G. So that's how you complete this table. The next part at the bottom, very similar to this, just slightly different format. I've given you a strand of DNA, both the template and the second strand or complementary strand. Here you're going to figure out, okay, so I need to use my template strand. So you're going to use this strand, you'll copy that down, and then you'll figure out what is your mRNA based on your template strand. Finally, the next piece is just a question asking you to summarize what you find. When you're done with those, make sure you just go on to Schoology and do your exit ticket. Other than that, please feel free to reach out if you have questions. Stay awesome, stay safe, and